Assalamu alaikum dear students, it's Dr. Masuma Mehtab. Welcome to my YouTube channel Medical School. So in this short tutorial, I'll be explaining granulomatous diseases of nose which are caused by bacteria. So let's go. Now the bacterial infection includes rhinoscleroma, syphilis, tuberculosis and leprosy. Okay, so starting with the rhinoscleroma. Rhinoscleroma is a chronic granulomatous disease which is caused by Klebsiella rhinoscleromatis or bacillus. Now it's endemic in several parts of the world like India. Okay. Now come to the stages. We have atrophic stage, granulomatous stage and cicatricial stage. So the atrophic stage resembles with the atrophic rhinitis. Now in this case what happens, there's foul smelling crust, purulent nasal discharge. So this stage resembles with the atrophic rhinitis. And then we have the granulomatous stage. As the name suggests, there will be granulomatous lesions or nodules in the nasal cavity, and there will be subdermal infiltration of the lower part of the external nose. Now remember here the nodules are painless and non-ulcerative. Now the third stage is a cicatricial stage where there will be stenosis of nares, which means narrowing of the external nares, and then distortion of upper lip and adhesions in the nose, nasopharynx and oropharynx. So here you can see this is the nodular lesion which is caused by Klebsiella rhinoscleromatis. As we have discussed, these nodules are actually painless and non-ulcerated. So the diagnosis after the clinical examination, you have to go for the lab diagnosis like biopsy. So the biopsy will reveal the presence of plasma cells, eosinophils, mucilac cells, and Russell bodies. Now these two cells are very important for the diagnostic feature. How? Let's see. Now, this is the mucilac cells and this is the Russell's body. So the mucal cells are basically large farm cells, which means they are macrophages having centrally placed nucleus and vacuolated cytoplasm. Now in the cytoplasm of this farm cell, you will get the causative bacilli. As in this case, we have the Klebsiella, so you will get Klebsiella in the farm cell cytoplasm. The second thing here is the Russell bodies. So the Russell bodies are basically inclusion bodies found in the plasma cells. Now these are homogeneous eosinophilic inclusion bodies. Inclusion bodies means aggregation of specific type of protein. So here there are immunoglobulins which are precipitated or aggregated as inclusion bodies. They occur as, let's read what's written here. They occur due to accumulation of immunoglobulins secreted by the plasma cells. So this is the diagnostic, these both cells are the diagnostic feature of this condition. Okay, the treatment involves antibiotics, so you have to prescribe the patient streptomycin and tetracycline together and steroids to reduce the fibrosis. So the second condition is syphilis. Now it is caused by Trypanosoma pallidum. It can be acquired, it can be congenital. So the acquired, in the case of acquired, you will get three stages. We have primary, secondary and tertiary stage. Now remember that in the tertiary stage, there will be deformity of nose. Okay, now initially there is gumma formed in the nasal septum. Later the septum is destroyed and the bridge of the nose collapses, giving it a saddle nose appearance. Now here you can see initially there was gumma formed in the nasal septum until and unless the septa is completely destroyed. Now we know that the septa contains the bony part and the cartilaginous part. So the bony part is here and the cartilaginous part is here. So both of these parts are completely destroyed, giving a saddle nose appearance. So the treatment after the diagnosis by means of biopsy, you have to treat the patient with penicillin and remove the nasal crust by nasal irrigation. Now, nasal irrigation is actually a process in which you have to put some saline solution in one nostril and it has to leave from another nostril. That's a process to remove the crust. Then come to the tuberculosis. Now, it occurred due to secondary due to lung tuberculosis. So it happens secondary to lung tuberculosis. Now, in this condition, what happens is anterior part of the nasal septum and anterior inferior turbinates are commonly involved. Firstly, it has nodular infiltration followed later by ulceration and perforation of the nasal septa. So, in this case, again, we have the perforation and ulceration of the nasal septa. And most commonly, which areas are involved? We have the part of the nasal septum and we have the anterior inferior turbinates, which are commonly involved. So here you can see we have the tuberculosis of nose, and this is the ulcerations and perforations of the nasal septa. Now, the third condition is the leprosy, which is caused by the Mycobacterium leprae. Now, the infection in this case starts from the nasal septum and inferior turbinate. Initially, there is excessive nasal discharge with red swollen mucosa. Later, this septa ulcerates and perforates. So, in previous slides, we have studied about the septa ulcerations and perforations. Again, we are studying about the septa ulcerations and perforations in the case of leprosy. Now, in this case, there will be depression of bridge destruction of the nasal spine. So here is a clinical presentation. Here initially the leprotic nose looks like this, but then we have studied about the leprosy. So in the case of lepromatous leprosy, the face becomes lion face. Now the nasal septum has completely destroyed the bony part as well as the cartilaginous part. So this is the nose, the leprosy affecting the nose. So diagnosis is mainly, we know, by means of biopsy of the nasal mucosa. Now biopsy will reveal the acid-fast leprae bacilli in the foamy cells. Now these foamy cells containing the leprae bacilli are called as the lepra cells. Now the treatment includes antibiotic like isoniazid and rifampin along with the dapsone. So this was all about the lecture. I hope you understood very well. Thank you. Allah.